the paper that we chose was on Talins and cell spreading. So cell spreading is sort of when a cell decides to latch on to a certain spot in the extracellular matrix. It sort of has to sort of increase the area with the extracellular matrix and create focal adhesions to sort of create that sort of bond to keep it sort of stuck to the extracellular matrix. So focal adhesions, we've talked about that in class before. This is sort of, I stole this from one of your slides actually, Tori, earlier in the semester. Just a diagram on what makes up a focal adhesion. So we've got the fibronectin in the ECM. We have integrin poking out of the cell, connecting to the fibronectin. And so this goes, through the cellular membrane. And then we have, can you guys see my mouse?
Yeah, we can see your mouse.
Cool. And so then we have, several different types of scaffolding proteins that connect the integrin with the actin cytoskeleton network. In this paper, we're looking specifically at Talin, so one of the scaffold proteins.
And of course, whenever you're looking at anything in biology, you want to make sure that it's actually relevant to something we care about. And the first place anyone ever looks is cancer, right? Well, as it turns out, this, like everything else under the sun, is relevant somehow to cancer, but it seems particularly relevant in terms of metastasis because, for a tumor cell to metastasize, that means it has to disadhere from wherever it normally exists, including among the other tumor cells, and then travel somewhere else. And then it has to re-adhere to that new surface so that it can form a new tumor elsewhere. Well, we're talking about adherence, so it seems pretty clear that it's at least something that could be involved. And since cancer cells have a tendency to hijack whatever is in sight to suit their needs, I'm sure that it is involved in at least some cases. And then, because cancer is the thing that's quoted by everything, we looked for a couple other examples, and another example we found is that leukocytes, white blood vessels, which are normally just patrolling the bloodstream, looking for problems that it needs to address. When it finds those problems, it needs to bind to the wall of the blood vessel, so then it can squeeze through it. And in this way also, cell adhesion and cell spreading are necessary for health and general biology to occur. And we're also people, and we chose this paper for a reason. For me, it was mostly just academic interest. I've always been interested in not only looking at a structure as it exists in a cell, but how that structure is first created. After all, these are very complicated cellular machines, and it's not like if you throw 20 different components together, it will form the machine perfectly on its own. So I was very interested in how they were actually looking at the stepwise way that the cells build the focal adhesions.
Yeah, and so for me, similarly, I did have the academic interest as well, but I also had sort of a more personal connection. And as Aaron said, everything in biology is connected to cancer, and so is this. And I actually, when I was 16, I was actually diagnosed with cancer. So if that had metastasized, that would have sucked. But anyway, now that I have dropped that bombshell, let's move on into the paper. Yeah. Anyway. So the first thing the authors decided to do was knock out Talin and just sort of look visually how that affects cell spreading. So we have two Talins in humans, Talin 1 and Talin 2. So for this paper, the authors actually treat them as interchangeable. So what they did is they created Talin 1 knockouts. And then on those knockouts, they either treated them with Talin 2 siRNA or with sort of like a control RNA. And so in the control cells, you have within like sort of 30 minutes, you see, within 30 minutes, you see.
Can I ask just a general question? When did this paper come out? I missed that.
2008.
2008.
2008. Okay, cool.
Yeah,
so this is, okay, cool. Thank you. I knew it.
So yeah, within 30 minutes, the cell sort of spreads out into and flattens out into sort of a very clearly unrounded shape, sort of a taking on a certain morphology. These are, by the way, are human embryonic stem cells, I believe. And if you don't have any Talin, though, So it knocked out Talin 1 and Talin 2 siRNA. The cell sort of tries to spread initially, but then it kind of gives up and then sort of just balls back up into just what sort of was originally. So it's not really spread onto the surface of the ECM at all.
Did they watch the cells longer to see if they kind of like oscillate? Do they try to spread again?
I believe that they did watch it longer. They didn't say anywhere else, but for some of the other experiments, they have a much longer time frame. And yeah, I don't think they ever saw any respreading.
Cool.
Yeah,
I was just curious
never
about
mentioned
that.
it for sure.
Okay.
Yeah.
So E here is sort of a quantification of C and D. So in the control cells, we see the vast majority of them are sort of elongated into sort of a final shape. The Tailin 2 siRNA cells, so the biggest chunk of them were actually sort of in the state we see here at like 10 minutes or 15 minutes, where it's sort of spread, but it's still rounded. And then some of these cells are sort of able to go on to like the next stages of cell spreading, but the vast majority do not make it to the final sort of elongated shape. So after this, the authors tried to look at how knocking out Talin will affect focal adhesion formation. So to do that, they stained for Talin and for vinculin. So here, you can sort of see the focal adhesions, which is sort of the yellow spots where you have both Talin 2 and vinculin. They're forming at the edge of the cell, near the cell membrane. If you knock out Talin 2, naturally you don't see much of the Talin, but you also see that you don't have vinculin coming up at the edge of the cell anymore. You're not forming those focal adhesions anymore. So that shows that you need Talin to form those focal adhesions. And then they sort of zoomed in on the edge of the cell as it's trying to spread out. And hold on, sorry. There's like a bar on the side of my screen. Okay. So they look at the edge of the cell, and you can sort of see here, there's this bulge sort of coming out from the cell. And it bulges out, and it's able to keep that bulge out. It's able to sort of keep spread. Whereas for J, you can actually see this whole sort of thing is the cell tried to spread out this whole thing here, but it's not able to keep spread out, and it recedes back in towards the center of the cell.
So I just have some questions about this. I think this is really interesting looking data. I like little montages like this to look at and kind of graphs and stuff. Sorry, not sorry. So they're just looking at GFP. They're just expressing GFP in these. And what's interesting, like you can see how thin that is. So I'm guessing in those bottom ones, this is just my eye looking at these. Like since there's no GFP signal, you see how there's kind of like that, there's that boundary of GFP at the membrane, and then there's like that clump of it cytoplasmically. That makes me think that those sections of membrane where it's dark there, they're basically fully collapsed onto each other. And so there's no real, there's a little thin layer of cytoplasm in those sections, but then it's like thicker on either end. It's like got this little ripple.
Yeah.
But then it's like actually stuck down to the glass there. So talins and like focal adhesions are actually like kind of important, it seems like, for like keeping the cells up and off of like the glass slide. Otherwise they kind of like fall down like little like clams or something,
Yeah.
It gives them their skeleton to stand up.
Yeah, and it also draws the cytoskeleton to the edge of the cell. So, if you don't have that, then it's possible that the cytoskeleton just never actually expands like the cell necessarily wants it, or at least not as much.
Yeah, so what is happening, and then it almost looks like it's fully collapsing by like 11 minutes all the way out there. Like there still would be membrane there, and you can kind of almost, I don't know, it's hard to tell.
I think the actual membrane is sort of coming in towards the center.
Yeah, I wish we could kind of see that better. The DIC is hard to tell where the outline really is.
Yeah.
It probably would have been good if they used something.
EM. I wish I had some EM. I wish I had this image with EM, and then we would be able to know.
Oh yeah.
Or
Yeah.
maybe some label other than cytosolic GFP that's just floating around.
Yeah, like membrane, like an outside membrane marker or something
Yeah.
could
Yeah.
work. Yeah. Cool. Thank you for
Sure.
get in there.
All right.
Yeah. So the next thing they decided to look at is they wanted to see what contribution the different regions of the Talin proteins had. So they cut Talin into two pieces, the N-terminal end, which they labeled the head of Talin, and then the C-terminal end, which they labeled the tail. And if you look over on the left in figure 3A, you can see that when they add in GFP Talin 1 head, you actually can rescue the semi-spread state that normally isn't present at all if you have no Talin. But when you add the Talin 1 tail, there's no change from the balled up state that Talinless cells always end up in.
Which end does the head bind to? Does it bind to the integrin or to the actin?
So if that's actually what they were looking at in FG and H, it actually binds to the integrin. And what they found is that the head region of Talin alone can actually bind to and activate the integrin, causing it to go to the edge of the cell membrane.
Okay, that's this paper. Okay, cool.
Yeah.
Thank you.
So in F, this is just with normal Talin. You can see that the GFP Talin is on the outer edge of the cell membrane. There's a lot of beta-1 integrin on the outer edge of the cell membrane, and F-actin forms the ring with stress fibers reaching out to the focal adhesions formed on the edge of the cell membrane. When you, in the middle frames, when you have just Talin 1's head, you can see that GFP Talin 1 and the beta-1 integrin are still at the edge of the membrane, even as the actin cytoskeleton is not connecting to it.
Yeah, that's. I would like to go back on this data.
Go ahead.
I just would like to compliment. You know, I do a lot of shitting on, but I would really like to compliment this FGH figure here. This is like art to me. This is it's not only very clear and good data, but it's beautifully presented. And I just want, I mean, it's a little, there's a little misalignments here you can kind of see, but other than that, it's very nice. I love the way they've done this merge with the zoom, and then we have these nice panels. It's pretty.
I'm also very glad they had unmerged panels so that you can see a little better what's going on with each of them.
Yeah, and they're very clear, and like the contrast is good. Like this, you know, these morphological differences here are also very fascinating. Like, you know, we could get in here and do some modeling on these different structures and tensions just by kind of looking at these cytoskeleton structures. We could model kind of like the behaviors of these cells and things with computers now, I think. That would have been a lot harder to do in 2008, I guess. Maybe not, I don't know. Okay.
So what they also found is that while the Talin 1 head can activate the beta-1 integrin and cause it to go to the edge of the cell membrane, it actually cannot cause the other parts of the focal adhesion complex to migrate. So you can see that in D, they looked at the localization of vinculin, and in E, they looked at the localization of paxillin. And in neither case could Talin 1 head cause the protein to go to the edge of the cell where it would need to be to actually form focal adhesions. In short, in figure 3, they found that the head domain of Talin 1 only can activate the beta-1 integrin, but can't recruit the other parts of the focal adhesion complex. And the net
I'm sorry. Yeah, sorry.
You're good. The net result of that is that having Talin 1 head alone actually allows the cell to remain in the semi-spread state they'd noted earlier. So here we're comparing figure 1C and D to figure 3C. And in figure 1, you can see that on top, the cell spreads normally over the course of about half an hour. But when you delete Talin, then you get some hesitant spreading followed by a loss of that spreading. But when you add Talin 1's head domain only to a Talin deletion cell, you get the cell begin to try to spread, and then it stops trying to spread, but it also doesn't retreat either. And by the end of an hour and a half, which is, I might note, 6 times longer than the last frame they showed partial cell spreading in Talin in Tailin deletion, you have still this partially spread state.
I mean, just how happy would you be in the microscope room when you did this experiment?
Oh, incredibly.
Yeah, it's just like I look at data like this and I'm just like, man, this would feel good. And it does. Like this just feels so good because we understand this now. You know, it's like we very clearly understand that Talin can activate these integrins and hold the cells down, but you know, we can't continue to spread past a certain point. It reaches a mechanical threshold or an expression threshold or something. Yeah, it's very beautiful. Very beautiful just data. You know, there's something to be said about like a nice clear paper and how impactful it can be. You know, the data is important, but the clearer it is too. You know, this is not even like meticulous data. Like these are, you know, different sizes and things like that. But like
you can see what's going on.
Yeah, it's very clear in what's happening.
All right. So after this, the authors decided to look at rearward flow. So what they did is they sort of created these alpha-actin and GFP mutants, and alpha-actin is one of the scaffolding proteins present in the actin cytoskeletal network itself. It's what allows the actin network to like form branches. So with this mutant, is sort of looking for they're sort of looking at the cytoskeleton. And so what they see is when you have sort of full Talin, like the cell will sort of push out and then it'll sort of flow back a little. The cytoskeleton will flow back a little. But then if you only have the tail in one head, this flow increases like a lot. So what they're sort of showing here is that when you have the focal adhesions present, that sort of helps prevent this rearward flow of the cytoskeleton network, where sort of it'll push out and it'll like form the focal adhesion. Then it'll recede back maybe a little bit, but it'll still have spread out to some extent. Whereas with tail in one head, it recedes right back.
Like increasing that zone of flow, like it's making the zone of flow bigger. Is that like because it's like making it wider?
Yeah.
Yeah.
And so then this is, but there's no pushing out happening here, right? Doesn't look like it.
Yeah, I think this is sort of like they watched the cell push out, and then once it had pushed out, they started measuring how much it flows back. I think that's what's happening.
But it's like it's just continually flowing, whereas these only flow after a push. They have like a push and then a spurt of flow.
I think that's
right.
What's happening. Yeah.
And then up here it's just kind of like flow, 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 flow.
I think so. They don't actually state at what time this is.
Okay.
Yeah, the way I interpreted this is that for both of these, it's just like, this is how much it flows after a single push, I think is what's happening.
Okay.
Yeah, something like that.
That's how I read it as.
Yeah, I mean, so the way I would read it as, so you see that right where they have that rearward flow and that arrow, that you see that membrane, you see that then it also goes up. And so that's the push. And then eventually that snaps back down a little bit. And you only get that flow after that push. Did you guys see that?
Oh,
does that
like this push here?
Yes, that is like the membrane protrusion.
Okay.
Does that make sense?
Yeah.
So then there's no pushing out happening down in that lower one that I really see, but it still has that strong flow. But it's not super clear that there's not flow happening up here to me either, because it looks like there's flow happening over on the left side, maybe.
Yeah, like there's some rearward flow basically.
All the time.
But
even
it's like a lot less is what they're saying.
Yeah.
Right. Like they managed to restrain the rearward flow to the small region near the membrane, where presumably the actin is still working on binding to the focal adhesions.
Yeah, right. So like Talin is like important for like constricting that flow zone. Yeah, that's cool. Okay.
Then they looked at the myosin light chain and F-actin networks. So in sort of when you have full length Talin present, you can sort of see these stress fibers forming in the F-actin network, these lines jutting out. And you can also see that you have myosin sort of localizing towards the edges of the cell. So like what this is sort of showing is that the myosin is trying to pull the actin back in, but since the actin is latched onto these focal adhesions, it's not able to do that. So then the myosin itself just sort of stays stuck there. It keeps trying to pull, but it's not able to. But if you only have the Talin 1 head, you can see that the myosin sort of is able to pull, and it pulls it all the way back towards like, near the nucleus, towards the side of the cell. And you see that you sort of lose the formation of the stress fibers that you had with full-length Talin. So yeah, the Talin 1 head alone isn't enough to sort of anchor the actin down and, sort of lead to the creation of these stress fibers and have myosin towards the edge of the cell and everything like that.
Sorry, can we go back? Yeah, this is really nice. I just wanted to say that this data is really nice, and I think that it's really interesting how the actin structures have changed there. I would really like to explore that, as a researcher. But I just wanted to say that the myosin light chain is smiling at us and like an upside down smiley face. Okay, that's all.
All right, cool.
Yeah. So, we needed to make a model, but we were faced with the conundrum that neither of us are any good at drawing or using online figure making software. So we ended up deciding to use Minecraft because, well, we both had some small amount of skill at manipulating things there, and also it was much more fun. So if you give me a second, I'll log on to Minecraft. And
I mean, is this like a dream come true for me that I get 2 Minecraft models in one day?
Let's see.
Ours doesn't have any creepers though.
Yeah, sadly, ours is just a visual model. It doesn't have any
You will get less points because there's no creepers. I'm sorry, that's just how it works. It's only fair. Creepers, they're out there, they're creeping.
Okay, give me a sec. Can you all see a Minecraft pause screen?
Yep.
Cool.
Yes.
So yeah, this is our visual model. So there are 6 panels here, and I'll walk through what each one is specifically, but basically we have 3 different conditions. And for each condition, we have a zoomed in molecular level model on the right, and then we have a zoomed out cellular level model on the left. And I apologize if it starts raining or the sun goes down because we haven't managed to get any control over that.
Oh, I'm sorry. That's all my fault. I also I also love that we just have all these sheep hanging out on our plasma membranes and shit. That makes me so happy.
Yes,
I had to kill a few to build these models, but anyway.
Yeah. But so what we have here is this yellow rock at the bottom is the extracellular matrix, and then glass is the plasma membrane. And you can see in orange the integrin that reaches through the plasma membrane and latches onto the extracellular matrix. And above that, in the wood of 2 different colors, that's all Talin. So the lighter colored wood is the head, and the darker colored wood is the tail. And together, those can reach up and grab onto some other cytoskeletal scaffolding proteins, which are not particularly important to define here. And then those can connect to the actin cytoskeleton, which is what the fence represents, which stretches out into the cell and connects this whole assemblage to the cell's overall cytoskeleton. And this is a wild type cell, so that's what normally happens. And when that happens, moving over here, you can see that the cell is just spread out pretty flat against the extracellular matrix, which is still the glowing yellow glowstone. Inside, because we made the plasma membrane out of black stained glass, you can see that there are focal adhesions where you have the orange integrin and the wooden Talin that together recruit the brick, which is all of the other proteins, that pin the cell to the extracellular matrix all around its perimeter. And that's the wild type model. Our next model is when you have a Talin 1 head only. So you can see that there are still the orange integrins that are bound to the extracellular matrix. And the integrins are still bound to the head of the Talin, but without the tail of the Talin, it can't recruit any of the other scaffolding proteins. And ultimately, the integrin head complex is not bound whatsoever to the actin cytoskeleton. So that's just kind of free floating up here away from the plasma membrane. And as a result, you get an intermediate phenotype. So in this model, both black and blue glass are the plasma membrane, but they denote different regions of the plasma membrane. So in black, you can see that the cell is rounding up. It's not particularly flat against the extracellular matrix. It's kind of just partially spherical as it existed before it tried to begin spreading. But in blue, you can see that there's a little bit of cell spreading that is pinned lightly by the integrin Talin head complex, but it doesn't have the Talin tail or the focal adhesions necessary to connect to the cytoskeleton, which would allow it to finish spreading. And then in our last model, we're just going to take a quick look at what happens if there's no Talin in the cell at all. Well, when there's no Talin, you don't get any integrin in the plasma membrane. There's all sorts of other proteins that we didn't bother putting in here, but there's no integrin. So you have no connection to the actin cytoskeleton, and basically you just have the cell sitting a little bit on top of the extracellular matrix. And the net result of this is that the cell is almost all spherical. I promise this is a sphere, guys. And there's just this small connection to the extracellular matrix where the cell is actually sitting on top, because inside you can see that there isn't any integrin actually connecting to the extracellular matrix.
Could we please go in all three of these cells?
Of course.
We can walk in, yeah.
Yeah, I would like to walk in these. And
The lanterns were just lighting, by the way. They don't mean anything.
Them. Well, I like them in there. I just kind of want to see what this feels like to be in the cell. So yeah, it's like a tall.
Yeah, it's tall.
Like,
it's mostly round.
Yeah, I wonder like what this does to the mechanical environment inside these cells. You know, like think about that. Like that. Let's go to that other one now. Yeah. This one's basically a sphere, right?
Yeah, this one's basically a sphere that I wanted to try to not just have like a 2-block connection to the bottom.
Yeah, you did a good, I think you did a nice job like making like a rounded cell, you know, with these three, as someone who's made a lot of like dome structures in Minecraft, it's hard.
It's so painful.
It's painful because it's just like, you gotta, it's like doing knitting or something. You gotta always be paying attention.
Yeah.
Yeah.
So yeah, this
one too. This boy here is also not any small feat to make.
Yeah.
We were briefly considering making square cells to represent round, but we thought it would be too much of a meme.
Yeah, no, I like that you took the time to make them round. This one feels like a dance hall or something.
Kind of. I'll be honest, I've thought about replicating these in a Minecraft world just as a house.
Yeah, I like this. This feels like a desert dome home, like made out of mud or something to me. I like it.
Mm-hmm. Oh, by the way, these are just on the Class Creative server, and they have little labels on them for what each type of block represents, so people can come back and take a look at them as they wish.
Cool. What kind of direction are they in? Northeast, southwest?
They're right next to spawn.
Right next.
Yeah, they're pretty close.
You literally cannot miss it.
Great. And then let's go to that last cell. I'd like to.
Yeah. I hope it's not too laggy. I'm trying to keep movement to a minimum.
So like what this has shown to me, like I'm glad that I did this Minecraft stuff with you guys and that you guys were on board with it. Because like I did that one little model where I was able to like model migration, like do an actual mathematical kind of model. And then Katie's was able to do an animation, a 3-dimensional animation that caused this, you know, bulb that she was seeing on these neurons. And then here we were able to see 3 different 3-dimensional molecular models that we designed and we can walk through. So it's the versatility here is amazing.
Yeah.
I'm a big fan of this as a tool now, teaching tool. Okay.
Not gonna lie, it was also way more fun than drawing would have been.
And I'm sure you're gonna remember this forever.
Yeah.
What a
way to end my semester.
You're gonna remember Talin forever. Yeah, I'm glad. I'm very happy, and I'm gonna put all these online. So I'll let you take back over the control of everything.
Cool. I'll let Humza grab the screen share back.
Yeah. All right. Cool. So we've seen the model, and all right. So just a quick summary, I guess. You need Talin to form focal adhesions. Talin is used to link integrin with the actual cytoskeleton. So the head region of Talin is the part that binds to the integrin. And if you don't have the tail, you can't bind to the cytoskeleton, which means you can't really form a focal adhesion, and you can't really have cell spreading, like only a limited amount. And you actually need cell spreading to happen for the cells to attain their proper shape. And then, so for our discussion activity, for all 6 of us here, 5 of us.
Hey, no, a number is just a number. I'm excited for this.
So let's imagine we've all gone camping, and we have to keep our food safe from bears. So we have to put it in a bag and then hang the bag off a branch like 15 feet in the air. Okay, and we have different weights of rope. And the heaviest rope is the only one that's like strong enough to actually keep the bag up there. But it's too heavy for us to like throw over the branch like 20 feet up. So how would you go about getting that bag up there 20 feet up?
One other thing, bears can kind of climb trees, and also raccoons can too. So this has to be suspended away from the trunk of the tree.
Yeah.
Like by at least 5 feet, preferably 10.
I have ideas, but I will let others go. Lana or Katie, do we have? We can just all talk. There's just the 5 of us, so we can all just.
Yeah.
Yeah, sorry to put the like 3 remaining people on the spot for this. This was originally planned to have like 20 people.
Hey, no, it's my fault because it's just, I mean, it's nobody's fault, but it's just here we are. I'm into it. If people, I just don't want to take. I mean, I can say it, but I'm the dang teacher. I don't need to say it.
Like, if the bag is too heavy to throw over a limb, can you just throw like the rope over the limb?
The rope itself is too heavy.
Yeah.
Oh, okay.
Like, remember, you're trying to throw it 20 feet in the air at least to get a branch high enough to haul the bag up 15 feet.
Right.
Are we trying to like create like a network? So like using the thinner ropes to get the thicker rope to be the thing that holds up the bags? Like, does that make sense? I don't know what I'm saying.
Yeah. No, that's actually pretty close to what we were thinking. So the idea is you can throw the thin rope. It's maneuverable enough to throw, but the thin rope isn't strong enough to hold. So you throw the thin rope over and use that to haul the thick rope up. And then at that point, you can use the thick rope to tie the bag up in the tree, but you never actually had to throw the thick rope over the branch.
That's a great idea. Do you want to hear my idea?
Sure.
I'd love to.
All right. I'm gonna get some mud, paint my face. I'm gonna take all these ropes. I'm gonna go out in the woods, choke all the bears.
Oh God. Well, that's a little bit unorthodox, but I suppose it theoretically works.
No, I think
that
that was a great discussion question. It's a really interesting system because like a lot of people would get this, they have this problem presented to them. They're like, oh, there's no solution. So I like that. It's like thinking outside of the box. And now I'm curious to see how you tie it into the cells.
Yeah. So this discussion activity is also our analogy, where sort of like the thin rope is sort of like our integrin. So it's sort of used to like the cell sort of initially uses it to bind to the extracellular matrix, but it's not strong enough to keep the cell, like to get the cell to really spread. Only just like a limited amount of spreading, I guess. And so to sort of keep spread out and do the actual heavy job of having, the cell take on its full elongated shape, you need the heavy rope. So the heavy rope is sort of like the focal adhesion and like the stress fibers. So yeah, that's our analogy.
Yeah, that's great. Yeah, that's a great analogy and a fun little. I like that discussion activity. It was more just like a thinking problem to get in this world rather than at the cellular world. That was I like that. That's fun. I'm definitely gonna steal that because it's easier for people to jump into those thoughts because we're familiar with this world. So
Sure.
that's a nice thing that I learned there from y'all. Yeah, great.
So, if there's any questions.
I don't have any.
Those were really awesome.
Thanks.
Thanks.
Yeah, I mean, all of your presentations were awesome. So thank you for that. That was a lot of fun. The two Minecraft models were amazing. I cannot. My heart is soaring to the planet and back. I can't. Yeah, it's just there. I would have never thought to make an animated 3-dimensional model, Katie. I don't know why, but that is just mind blowing to me. And then
Yeah.
the fact that you guys made the 3 and that we could go in them, it was beautiful. I was like, I'm gonna make all my scientific models in Minecraft now, like my real ones that are important. Because why wouldn't you?
Yeah.
Because then you could take little videos and be in there.